isopycnic cesium chloride gradient sets up. Takes about 20 to 24 hours for it to set up. So this is the main reason why we have two sections, is that we don't have class on Tuesday and Wednesday and Thursday. So we started on Tuesday and take it down on Thursday, and we only have one ultracentrifuge. So that's why we had to offset everyone's experiment days. Okay, so you have this differing density gradient that can separate the protein from the RNA from the DNA. But we have a problem. We have two different kinds of DNA. We have plasmid DNA and we have broken genomic DNA. If it's intact genomic DNA, remember it's down with the pellet. So we don't have any intact genomic DNA in our sample. Only broken shorter pieces, but they shorter relative to the length of the entire E. coli genome is still pretty long. They're larger than pieces of plasmid, in other words. And if you have any plasmids that happen to have gotten nicked in their DNA along the way so that they're no longer a covalently closed circle, that can also be separated out with the two different kinds of DNA. So basically, we want to be able to separate our plasmid DNA, plasmid that's present as a covalently closed circle, versus plasmid that's been nicked and just broken genomic DNA. That we want to sequester those things together. So what we're going to do is we're going to add ethidium bromide. Ethidium bromide is a flat, planar molecule that is a very organic molecule and does not like being in an aqueous environment if it has a choice. It will, to a small extent, dissolve into an aqueous solution, but given the choice, it really, really wants to be in an organic environment. Now, when you are doing a prep like this, the organic environments it's going to try to sequester into are proteins, RNA, and DNA. And so proteins have an organic environment, so some of the ethidium bromide is going to bind proteins, and so your proteins will actually sort of make like a little solid ribbon of protein in the tube that, because you have it in a vertical tube, and because it's solid, it's the one thing in the tube that does not just shift seamlessly to be top versus bottom. So you may still see this ribbon of solid material, which is your proteins that have complexed with ethidium bromide, and just avoid, when you're trying to extract your band, just avoid that protein ethidium bromide ribbon. Ethidium bromide will try to intercalate into RNA to a small extent, but remember that RNA is single-stranded versus DNA being double-stranded. So it's not as happy in RNA. It does not intercalate in RNA nearly as much as it's going to intercalate into DNA. It really, really likes the environment inside of DNA. Inside of that double strand of DNA is a very organic environment. So there's a driving force for the ethidium bromide to intercalate between the bands of the RNA, between the bases of the RNA. So when ethidium bromide intercalates into those bases, remember that DNA is made up of all these flat planar molecules, and ethidium bromide itself is a flat planar molecule. So it's going to sort of wedge its way in there. But the problem is ethidium bromide can't quite fit, but it really wedges its way in there anyway. So when it wedges its way in between those bases of the DNA, it'll untwist the DNA a little bit. So that driving force to get it in there is so great that it will actually cause the DNA to untwist a little bit as it gets in between the bases of the DNA. So if you have, this is show and tell time here, if you have broken genomic DNA, so this is broken genomic DNA, it was as close as I could get. Oops. as the ethidium bromide intercalates and it untwists, it'll untwist here and it'll untwist here, and some intercalates here and some intercalates here and some intercalates here and some intercalates here. And you can see that it doesn't matter. You can intercalate a whole bunch in here because the ends are free. They're not attached to each other, and so they can basically freely rotate. So you can intercalate a whole bunch of ethidium bromide into broken genomic DNA. Well, as the ethidium bromide intercalates into the broken genomic DNA and untwists it a little bit, now it's taking up a greater volume. And when basically the same molar mass takes up a greater volume, the density decreases, Chem 100. So now you have approximately the same number of milligrams of DNA, and it's greater in terms of its number of milliliters. If you think about it that way, we're not actually measuring it, but it's taken up a greater volume. So the density of the genomic DNA is significantly decreased compared to any other DNA that's in the tube. Well, what about plasmid DNA, you might wonder? Okay, so plasmid DNA also has bases of DNA. It can also intercalate all this ethidium bromide in between the bases of DNA, and there's that same driving force to try to get it into the bases of DNA for plasmid DNA. But the difference is that plasmid is a covalently closed circle. And because it's a covalently closed circle, when ethidium bromide intercalates in here and it untwists a little bit, the rest of the molecule twists a little bit more. And it intercalates here and it untwists, and the rest of the molecule twists a little bit more, until pretty soon it's intercalated so much that the rest of the molecule is all twisted up and actually cannot incorporate anymore. So while broken genomic DNA, because of its free ends, is able to intercalate a lot of ethidium bromide and significantly change the density of that DNA, plasmid DNA, because it's a covalently closed circle with fixed ends, it is not able to incorporate that much ethidium bromide, and the density will only change very, very slightly. So if this is, say, 1.6 right here, plasmid DNA goes up a tiny bit, but genomic DNA goes up a significant difference. And so now, because we've added ethidium bromide to our solution of all of our things, now we're able to separate the genomic DNA and any nicked plasmid DNA, which is no longer a covalently closed circle, from the covalently closed circle plasmid DNA, which is our goal. And the other benefit is we can now see it under ultraviolet light, because under ultraviolet light, ethidium bromide will fluoresce into a visible wavelength. So we'll do this in the dark, and you'll actually see the fluorescence, and you'll be pulling out, you'll see the different layers of DNA. So you'll see the RNA at the very bottom of the tube, you'll see the protein, which will either be the top or like in a little ribbon, and you'll see the two layers of the plasmid DNA, and the plasmid will be below, and the genomic will be above. And so we will want to separate out our plasmid DNA. So just as a reminder, it's actually being set up, it's being spun vertically in a vertical rotor. Anything that's liquid, as the rotor slows down and stops, it just sort of goes, and so what's outermost is then at the bottom, so that would be the RNA, and what's innermost, which would be the protein, is now on top, and so it is going to look like that when you see it in the tube. So then we will want to be able to selectively get just our plasmid DNA out of the tube. So we're going to use a needle, and I'm going to be showing you this step, because in this case a picture really is worth 1,000 words of explanation. And so basically we're going to have it under ultraviolet light, we're going to see our band, we're going to put the needle in a specific fashion, which I will be showing you, such that the the beveled edge of the needle is just below the layer of the plasmid DNA, because it always sucks in from what's right on top of it. So you wouldn't want to, say, put it in the middle of the band, because it's going to be sucking in from right here, and you won't be getting the yield. So you always want it to be hopefully in the center. I drew the picture a little off, but just below the band, and you'll be getting your band of plasmid DNA. Now the genomic DNA, remember that genomic DNA is still very, very long, thick, and goopy. So you'll want to avoid getting any of that genomic DNA in your syringe when you're trying to draw in the plasmid DNA. Sometimes it happens. Sometimes people start to draw in genomic DNA. If that happens, you'll very often sort of see the genomic DNA layer sort of move like that. It'll sort of do what I call the wave. It'll sort of go, as you catch some of the stringy, long tendrils that might be hanging down in odd places in the solution, and you start drawing them in. It's like having a rope with a big old knot in it. You're trying to draw it in, thin string by thin string, but you end up with this knot of DNA basically. When you draw that in, you'll generally notice it will start to get very, very difficult to draw solution into the syringe. Another reason it might be difficult is when the very beginning, if you accidentally get like a little piece of plastic when you're drilling the syringe through the tube, that can also make it difficult. But that'll be difficult from the get-go, not from like halfway through. And so then when you start bringing in the genomic DNA, you'll be bringing in about 50-50 genomic and plasmid. And so then I'll be reminding you of this. You'll have a choice of, do I continue, do I stop? Because the second spin we're going to do, you will also be able to have a second chance at isolating it. So you're going to be extracting this band. You're going to be putting it into a second tube. And in that second tube, you're also going to be spinning it again, setting up its own gradient, and again, going in with a syringe to extract it a second time. Has anyone in here ever used one of those DNA isolation kits, like from QIAGEN or something like that? Yes? Yeah, a couple of you? Okay. So if you look at the original big instruction booklet that came with it, which is probably just online right now, one of the things it traditionally says in there, it says, gets the DNA as clean as 2x cesium chloride. And so that's what this procedure is two times cesium chloride gradient. So when you use the QIAGEN kit, it's saying, we have very high quality where we're getting DNA to the same level of clean as doing this procedure. So why does everyone use a QIAGEN kit now instead of doing this procedure? Because it's a lot faster, and it generally will get it as clean as. So we're showing you this technique not because we think you're going to use DNA, not because we think you're going to isolate DNA using this technique, but density gradients are very useful for a number of other things. For example, Dr. Pullman in his lab, when he needed to isolate some silver nanoparticles, he drew in some students from this class who'd done density gradients and who understood how density gradients worked, and he used that to isolate his silver nanoparticles. So basically, remember what we're teaching you in this class is a series of techniques that you can mix and match for other reasons outside of, and we don't ever expect that you'll do this exact procedure to isolate DNA, just as we don't ever expect that you'll truly ever isolate aldolase or do, LD, or need to do LDH kinetics, because frankly, LDH is pretty well characterized. But what we're teaching you in this class is a series of procedures that you're going to be able to mix and match because you know the theoretical background behind why you're doing each of these steps. And that's what makes it so useful to people going to work in industry, that you've learned how to think about what you're doing, and that you know the chemical basis for all of these things you're doing, and you know how to troubleshoot your experiments, which is why we have the emphasis on error analysis. So this experiment is just showing you how to run a density gradient, basically. Okay, so after you've done this twice and you've collected your sample for the second time, your DNA has ethidium bromide still intercalated into it. So we need to get that ethidium bromide out of the DNA, because if it's left in the DNA, it's going to eventually nick the DNA. It's putting the DNA under some torsional stress and making it much more likely to become nicked. And we want our DNA to be maintained as a covalently closed circle if at all possible. So you're going to extract the DNA with isoamyl alcohol. Isoamyl alcohol, and you'll remember your organic chemistry class. You did extractions in organic chemistry, yes? I mean, during COVID, hopefully you did something like that still. But basically you're mixing together with your aqueous solution containing your DNA, you're mixing an organic compound, isoamyl alcohol, and the ethidium bromide prefers the organic environment of the isoamyl alcohol. And you just have to vortex it together and then spin to separate the two layers. And you'll see the top layer is pink because now the ethidium bromide has come out of the DNA and gone into the upper organic layer, and then the lower layer, which is the aqueous layer, and contains the DNA, is less pink than it was. And you'll extract at least twice. After you do your second extraction, if your aqueous layer, where your DNA is colorless, you don't have to do a third one. But if you still have some color in your DNA layer, you need to get rid of that ethidium bromide, so then you would do a third extraction. So at least 2, maybe 3 extractions with isoamyl alcohol. And then your DNA is going to be in, maybe 5 mils volume, 2 mils to 5 mils of volume. Well, you need to do a couple things. It currently, it's just out of a cesium chloride gradient, so it has a significant amount of cesium chloride salt in it. Cesium chloride can be an inhibiting factor in later uses of the DNA. If you are trying to do a restriction enzyme digestion, it can inhibit restriction enzyme digestion. If you're trying to take some of your DNA and get it into tissue culture cells, it can impact the tissue culture cells, because basically it's a heavy metal salt, and tissue culture cells and a lot of enzymes don't like that. So we need to do a couple things next. We need to get rid of the cesium in the solution, and we need to get our DNA into a consistent volume where we can measure the concentration of it. So we're going to do 2 ethanol precipitations of our DNA solution. And the first thing we're going to do is bring it up to a larger volume, and usually rinsing it out with some water from the original tube. So you'd have a tube here where you did the isoamyl alcohol extraction. You're going to take that, dump it into a 50 ml tube, rinse it out with some water so you make sure you're maximizing your yield of your DNA. Dump that into the original tube. Everyone's going to bring it then up to 15 mils just so everyone has a consistent volume. And also because if it's too concentrated, your cesium chloride salt will tend to precipitate out, as well as it tends to precipitate out at extremely cold temperatures like -70. So we're going to be giving it a salt to replace the cesium. Can anybody tell me why DNA might need salt to efficiently precipitate? What's on the backbone of DNA?
Phosphates.
Phosphate. What's the charge on phosphate?
Negative.
Negative. So it's going to need some cation to be able to precipitate down. Now, if cesium's all it has, sure, it's going to use cesium. But cesium's a very large molecule, and we're going to offer it the substitute of a very small molecule. So we're going to put some sodium acetate in there. And so it prefers the sodium, so it will be better able to precipitate efficiently with sodium than with cesium. And we're also going to add ethanol, which is why it's called an ethanol precipitation. And the reason you put ethanol in your DNA is DNA has a minimal solubility, And you guys you had organic chemistry, so you're familiar with like minimal boiling azeotropes and maximum boiling azeotropes and things like that from organic chemistry? Somewhat? Okay. So DNA has minimum solubility at approximately 66 to 70% ethanol. So if you have an aqueous solution and you put in 2 volumes of ethanol, you're basically making it 66% ethanol, and your DNA will precipitate out. And your DNA will precipitate out no matter what temperature it is. It will precipitate out at room temperature, but you can get a little bit better yield by putting it at a cold temperature, an extra couple percent yield by putting it at a cold temperature. But remember I said there's still cesium salt in there, in the solution. It's not with the DNA. The DNA has the sodium with it, but you don't want that cesium to also be precipitating out. We're trying to get rid of that cesium salt. So if you put your DNA solution at -70 or in an ethanol dry ice bath to precipitate out, the cesium is also going to precipitate out and you can't separate the two. But if you put it only at -20, the cesium will stay in solution, your DNA will precipitate out, including that slightly better yield. It's cold enough to get yourself a better yield of the DNA, but it takes longer. So you put it at -20, you have to put it at -20 at least overnight if you're doing this in lab. If you needed to ever precipitate out your DNA quickly, you stick it into an ethanol dry ice bath, which is what we're going to do the second time. And when you stick it into an ethanol dry ice bath for 20 minutes, boom, you have maximum precipitation. But we can't do that yet because we still have cesium in there. So the first precipitation, we get rid of the cesium when we pour off the solution, and we just have our tiny, tiny little DNA pellet at the bottom. And it will be a very tiny DNA pellet at the bottom. It'll be so small, you're going to be like, I'm not even sure I can see that sometimes. So I'll be showing you how to efficiently pour off your solution. One of the things I say when you have a pellet at the bottom of the tube and you have a solution on top of it, the worst thing in the world you can do is back and forth. You never ever want to do back and forth, because what you're doing is you're dislodging the pellet when you do that. You have to do what I call commit. So you have to just say, keep your eye on the pellet, turn upside down to pour all that solution off, keeping your eye on the pellet. That pellet starts to go anywhere, okay, then you can quickly, turn it right side up, but failing that, as long as that pellet's stuck on the bottom, you need to just commit and pour the solution off. Never ever do this, because guarantee it's going to dislodge your pellet if you are doing that. Okay, so you've added the sodium acetate, you've added the ethanol, and you've gotten your first pellet. Then we're going to re-precipitate out our DNA with cleaner, better ethanol. The first time we're using like 95% ethanol, which has some methanol, organics and stuff like that in it, and that's what we're getting rid of all that stuff. Then the second time we're going to use really, really high quality 100% ethanol. Those of you who have taken organic chemistry, do you know anything about ethanol and water in terms of a minimum boiling azeotrope? That was weird. Do you know anything about ethanol and water in terms of a minimal boiling azeotrope? If you just leave ethanol out open to the air, what percent ethanol does it tend to become? No one? So if you just have ethanol exposed to the air, it tends to become 95% ethanol. It will grab moisture from the air until it is 95% ethanol. So to get 100% ethanol, you can't do a distillation to get 100% ethanol, because it is a minimum boiling azeotrope. So they take their 95% ethanol and they have to add something like Dry-Rite to it to suck all the liquid out of there. So the 100% ethanol has to be specially purchased. It's very expensive, it's very regulated, and average people don't get to purchase it at all. You have to be a school. If I wanted to purchase it, I could not purchase it. You have to have an alcohol license as a school to be able to get the 100% ethanol. And that's because if you drink 100% ethanol, it's going to burn your throat. And I don't mean the nice burn that, you know, a nice whiskey does or anything like that. I mean, it'll suck the moisture out of your throat, and you'll actually have like burns in your throat from the 100% ethanol going down. So for that reason, we do not encourage anyone to ever drink 100% ethanol. Not that I'm going to give you the chance. Okay, so we've done the first precipitation. We're going to bring it up in a little bit of water. We're going to add sodium acetate and ethanol again, do a second precipitation, this time in an ethanol dry ice bath, very, very cold, for 20 minutes, spin it again. And this is all in your protocol, by the way. So if you need to follow along your protocol, spin it again, and then we'll have a pellet, and we're going to bring it up in TE buffer. And TE buffer is just tris and EDTA. Tris to maintain a pH, and just a small amount of EDTA in there, just to make sure that we are able to discourage any DNases, where we're chelating those divalent cations for, you know, the few that might be in the solution, so that any DNases would not have good activity, so that we can store our DNA. Ideally, you store DNA in the freezer, but if you're going to be taking samples of it a lot, then you would defrost some and put it in the refrigerator. Anytime you have a frozen sample, you damage your sample a little bit every time you defrost it. So if you think you're going to be using, say, a significant amount in 2 weeks or maybe 3 weeks, you would have that amount, if at all possible, in the refrigerator, and then keep the rest frozen. So you don't ever want to, say, defrost a sample every day, or, you know, in the morning and then again in the evening, because you're incurring damage every time you do that. Probably more than the damage that's going to be kept from, that's going to be accrued from just keeping it in the refrigerator for a couple weeks. So just things to keep in mind when you are working in industry in the future, working with DNA samples or something like that. Now, protein is another story. You may have no choice but to keep your protein maybe at liquid nitrogen or at -70. I know in Dr. Stump's lab, as soon as we were through working with the protein, it had to go back in -70 or would start to degrade very, very quickly. Okay, so that's pretty much it for this protocol. You're then going to be using a spectrophotometer, and you're going to be reading it at 4 different wavelengths. And I'm going to give, at the time you do this, a little more comprehensive lecture about each of those wavelengths and what numbers you should see and things like that. But basically, you're reading it at those wavelengths to be able to establish concentration and purity of your DNA. So those wavelengths will help you establish concentration and purity of your DNA, and we're going to be discussing how to, once you know the concentration, how are you going to get yield of your DNA from the procedure. And you can see the lab report, which is on page 79 of my lab manual. It's a pretty simple lab report for the DNA experiment. It's basically just your growth curve from your cells. You're going to be telling me the purity and the concentration and the yield of your DNA, showing calculations, error analysis, short and sweet, completely unlike your aldolase. Remember, I've been telling you all along, the aldolase is the absolute worst lab report you will do all semester, and it will get easier from now on. What gets every bit as picky, though, is your intros. And so for your intros, which Teresa is going to grade for me, because as you all know, as I've said multiple times, God, I hate grading. And since I'm retired, I don't have to anymore. So when you do your intros, whether you're doing the GST-SH2 intro, whether you're doing the plasmid intro, make sure you talk about all the solutions that are used. So if you're talking about the lysis buffer, in the plasmid experiment, we detail what's in the lysis buffer. It's basically the same lysis buffer for GST-SH2, same components. When you talk about lysozyme, for example, you don't just say, we're going to use lysozyme. You don't just say, we're going to use lysozyme to chew up the peptidoglycan wall, which is what it's used for, but you say, you know, we're going to be adding lysozyme. Lysozyme chews up the peptidoglycan wall. This is important because bacteria have a thick cell wall, and the membrane's not going to be accessible until the cell wall has some holes chewed in it, etc. However formal or informal you want to make the language, it has to be correct. Do some research on your own for these components. So just because maybe I've simplified it for a lecture does not mean that that's all we're expecting you to do. I may only say a couple sentences about it in lecture because I know that you all have the capability of doing research on your own, looking up lysozyme, seeing what it does, seeing how it works, seeing how it's going to apply to this experiment, things like that. So for every single chemical that you are using, make sure you talk about it. For every major technique that we are doing, make sure you talk about it. So for the plasmid prep experiment, make sure you talk about the cesium chloride density gradient. How does the density gradient work? How does the density gradient separate out all the components that you have from your supernatant, and how did you get that supernatant, from spinning, etc. So you're building that story. So you're saying, we're doing this, we end up with this, then we spin it and we end up with this, things like that. So you're telling a story, and as you're telling a story, you're talking about each of the components, the importance of the components in the reaction. If you're doing the GST-SH2 intro first, you want to make sure you talk about the glutathione beads and how the glutathione beads are going to be used to separate out the GST-SH2. And vice versa for both of you when you're doing the other assignments. So if you're doing plasmid prep first, it doesn't mean you should not be listening to what I'm saying about GST-SH2 either. So make sure you talk about how we're doing this differently from the aldolase experiment. In the aldolase experiment, we had a column and we put our stuff in the column and we ran it through the column. In the GST-SH2 experiment, we're going to be adding our beads, and then we're going to be spinning the tube down. And after some time, of inverting it to allow it to mix, of course, we're going to be spinning it down so our beads are at the bottom and removing all the supernatant from on top of the beads and adding the next solution and washing, things like that. So even though it's the same thought, the same basis, it's done differently. So make sure when you are doing those intros that you explain everything. Okay, so now intros are worth more than reports. Intros, I think, are 40 points and reports are only 35 points. So more work in the intros, less work in the reports. Okay, any questions about what you need to do for either the GST-SH2 or the plasmid report? And note that the plasmid report does not to be turned in until Thursday, because I still need to show you guys the autoclave that's on the third floor and explain to you how an autoclave works. And when you learn how the autoclave works for your particular experiment, and the GST people will get this lecture later, and the plasmid people will get this lecture on Tuesday, and that gets incorporated into your intro as well. Okay, any questions? Yes?
Plasmid's 2 days, right?
I'm sorry?
Or this lab is 2 days, or?
The plasmid lab is way more than 2 days. We tend to, because there will be days when you're just doing a little bit on the plasmid lab, we'll be doing a little bit on this experiment, and then we're going to be bringing in the PCR lab as well. So we're going to have overlapping experiments, kind of like real life. So if you're ever working in research in a lab or working in a company, you're not only doing one thing at a time. You might be working on this project, moving this project a little bit forward. Maybe there's 1 hour a day of work to do on this project and 3 or 4 hours a day of work to do on that project. So it's not at all uncommon to be working on several different things at one time. So in this part of the semester, we're showing you how you can do that. So we'll be working on, for the aldolase, we're showing you how multiple different parts come together to make one big report. And when they do plasmid and PCR, They're actually different experiments, but you're doing it at the same time to learn how you can be doing more than one thing at one time. Unlike probably many other, it's completely unlike Chem 200 or 201 or organic chemistry. I think for the most part, in all of your labs so far, you've just done this experiment and then this experiment and then only had to focus on one thing at a time. But real life's not like that. So we're giving you a little taste of that. Okay, more questions. I know, that was the 5-minute answer to a simple yes or no question, but you will find that I am incapable of just answering yes or no to a simple question. Always more information. If you don't have any more questions, you can go.